So we have with us Dr. Subramanian, and she is an associate professor of biology at Simpson College. And we are honored to have her here. And we'll just start by letting you talk about what you do, and then they are ribbon with questions.
Okay, great. So I'll not talk too much then because I bet they have a lot of questions. Um, so I've been at Simpson College for 6.5 years. I teach cell and molecular biology, which is everyone knows about biology, but biology is such a vast field. You could look at, the body as a whole. You could look at how the environment interacts with the body. You could also look inside the body, and my favorite part is looking inside the body and inside the teeny tiny cells that build all your organs, right? So you have cells that make up your heart, cells that make up your kidney, your brain, so on and so forth. I want to look at what's happening inside the cells. So it's like saying when you have a big city, right? You could study the entire city of Indianola, or you could study what's happening inside your houses, right? You'll still have a good understanding of what Indianola looks like if you know what's happening in your house, how many people stay in your house, where do they go to work, where do they go to play, so on and so forth. So I like looking inside the houses. That's how I like to explain that.
That's a great analogy. That's really neat. So we've just started getting into cells and talking about that. So yeah, I will just open it up for questions with them, if that's okay.
That's
Okay.
great. Yeah.
Oliver, you want to start nice and loud?
What's your first experiment that's failed?
So they want to know about failure. Yes. Talk
Oh my
about
God,
that.
there are so many. There are so many. Do you want to know after I came to Simpson or when I was doing science when I was young?
How about when you were young? I think that'd be awesome.
That would be amazing. I think it was chemistry lab.
You don't.
In chemistry lab, uh, I don't think you'd ever heard about titration, right? So there was this, we had to use this flask where you had to add chemicals. And at some point, the chemical was supposed to change color to pink. And I just kept adding these chemicals and I kept waiting and waiting and waiting. Everyone else in the class, their chemicals turned pink very, very quickly. And as I was waiting, it took me like 20 minutes. Everyone else was done with their experiment. And I realized I was adding water instead of acid. And, and the best part is the bottle which I was taking the solution from said water.
See? Oh, see? It happens. It happens. They watch you compare and fail all the time.
And so my teacher was like, you can't read? I'm like, I can read, but I picked the wrong bottle. So it was embarrassing, but you know what? Who cares?
Right. And you learn from it.
Yeah.
Lydia, question.
Why did you choose to study cells?
Oh my God, that's a great question, right? Like, it's easy for me to tell why I chose biology, but why cells? Okay. I don't know if this is going to make sense, but apart from biology, I also like art. And so when I started getting interested in like cell biology, I used to paint a lot. And sometimes when I used to look at pictures of cells under the microscope, they would look like paintings. And so to me, the more I do microscopy, the more I look at cells under the microscope and use different dyes and pretty colors, the more I see and the more I feel like I'm doing art when I'm doing science. And so to me, cell biology is like doing art. And so that's what attracted me to cell biology more than, I mean, I like all sorts of biology, but cells are more fun to me. No, maybe someone else likes something else, but to me, they're more fun than anything else.
That's beautiful. Josh?
What was your biggest discovery?
It's been your biggest discovery so far.
That is a very, very good question. Ooh, okay. This is very fun. So, right after I got my doctorate in cell biology, I was trying to get a full-time job, but I was doing a part-time job in an engineering lab that required biology with engineering. So, okay, I have to simplify this. What we had to do was we had to create fake bone, you know? So we all know what bones are made of, right? But I had to create fake bones and I had to grow cells on those fake bones so that we could take those things and say someone gets a knee injury or a hip injury. We could take those things, put it in the knees, and you allow the cells to grow and it fixes those knees or hips or whatever.
How cool is that, guys? That's
That is
Yeah,
so
and I did it for $10 an hour. I don't know, it won't make sense to you, but your teacher knows what $10 an hour means after getting a
for
pay.
such important work.
It's such important work. But the best part was it's very difficult to take cells and put them essentially on plastic and just tell them grow, right? But after like 2 or 3 months of being in the lab over and over again, I got the cells to grow on those fake bone tissues. And we got so many publications after that. I'm not even in that lab anymore, but it's been my favorite discovery.
That is awesome. Did they include you on the publications as an author? Because you did
Yeah,
that.
yeah, because I did all the work, so yeah.
You did all that. Yeah, absolutely. Yes.
What's your favorite experiment?
What's your favorite thing you've done?
Oh, my favorite experiment is what I'm doing now. So, right now I'm in Florida. You can't hear this, but I can hear the ocean right outside my window.
Wait, did you get snow though? Or no, where you are? Isn't Florida getting snow?
I'm not in that part. I'm on Cocoa Beach, where Kennedy Space Center is.
Wow. How cool is that, guys? That's awesome.
Yeah, I'm actually right next to Kennedy Space Center. I just did a presentation at Kennedy Space Center for a NASA project that I'll be working on. So my favorite experiment is, can I show you some of the parts? Hold on, I'll be back. Yeah.
Love that.
She's awesome. She is awesome. She's amazing.
And she gets to do her biology and that's so cool.
So I'm actually building this in my house. So look at this weird contraption. It's not fully fixed yet, and I'm not showing you the whole piece. But what I have to do is I have to take cells, put them in a beaker, put them on this rotating thing, right? And then you connect it to a computer and it rotates by itself. So what I like to call it is it's like a vomit comet for cells. Imagine if you were sitting here and you're just spinning around over and over again. So the idea is to grow these cells under what we call zero gravity. So on Earth, we have gravity, right? So we're all sitting or standing and we don't fall. But when you put them in this machine that we just 3D printed, what you do is you create a condition where the cells don't know what's happening and they're sort of in zero gravity. So it makes them think that they're in space. So what happens when these cells are in space? So studying cells will tell us what happens to our cells in space, right? Because all these astronauts, now they're going to the moon. They may go to the Mars. It might affect their cells, right?
Yeah,
So
absolutely.
so right now the experiment I'm doing with this, it's the coolest because if this experiment works, and fingers crossed, hope it works out. If this works, then I can send these cells to the moon in 2 years. If
Oh,
it works.
Wow. That is incredibly exciting.
Yeah, there's a rocket called Artemis going. I mean, I'm exaggerating. I'm simplifying this. There's something called Artemis which is going to the moon in 2 years. So if this works, I can put this on Artemis.
Oh, these stars. That's amazing. I'm just like thrilled that Simpson was able to get you. Like that's the things you're doing. Laurel.
so How did you become an assistant professor at Simpson?
Yeah, yes, I want to know this too.
Oh my God, you're going to laugh. So I was in Ohio before coming to Simpson. I was, I was in the microscopy facility. And one day I thought, I was bored. I was bored and I knew I had to apply to jobs because I was doing a lot of work, but I wanted to work with students. You know, at that point, I was just working by myself, doing my own research. But I really liked working with students, undergraduate students. And I wanted to become a professor because then I could teach students how to do research. After a point, you don't want to do research by yourself. You want to show people what research you're doing, how to let them become scientists, maybe. Or if they don't want to become scientists, at least teach them that they don't want to become scientists.
Yes,
Not
absolutely.
everyone has to become a scientist. So then I was like, let me apply to cell biology jobs. And I was just Googling cell biology jobs. The first job that came up was Simpson. I was like, okay, Iowa, it's probably nearby. It wasn't. But I applied to the job, and believe me, I didn't even show up for my interview on time. I missed my flight, and I showed up for my interview a week later. I didn't think they would even pick me. I'm like, I'm not going to get this job, but I'll still go. Um, I got the job.
That's a wonderful. Well, Mr. Remeyer, he's a pretty smart guy, so he probably knew what he was doing. I had Bryson, his son, a few years
Oh,
back.
nice. Yeah, yeah. I know Bryson. I know Bryson.
Oh, that's wonderful.
Yeah. So
Yeah.
I love Indianola.
Yeah.
Have you ever done like a really big project with it, like a bunch of people?
So yeah, like how many people do you usually work with or collaborate with on things?
Oh, okay, good question. When it comes to students at Simpson, I've worked with, let's say, 4 students at a time. This project I'm doing with NASA, there are a lot of people, but in different labs. So there are 2 people in Japan. There's 1 person in Canada. There are 2 people in the US. So in this project, I'm working with a lot of people, but they're not in one lab. But when I was in Ohio, in a university called Miami University, where I got my PhD, at one point, I was working with 25 undergraduates. That was a very difficult project because I had to I forgot who I told what to. It was a bit confusing in the beginning, but it worked out. So, yeah, working with 25 undergraduates in 1 year, that was the biggest.
It's a lot.
Yeah, yeah.
Okay. Scarlett?
When you were studying cells, like in different plants and animals, what do you find?
When you look at plants and animal cells, like is there anything that struck you?
Yeah. I have 2 or 3 different answers for that. These are all great questions and they're very complex questions. So good job, everyone. So plants obviously are different from animal cells. What I like about plant cells is when you look at them under different types of microscopes, they have such unique structures. In plants, I love to look at chloroplasts because they're so huge, and the plastids, as we call them, that give the chloroplast their color. They look very neat under the microscope. They look like these huge spherical structures. You can't miss them. They look very, very cool. And also because of their green color, when I use them under a different microscope, you can see the colors really well. And there's a lot, believe it or not, there's a lot of movement in plant cells. So there's this thing called cytoplasmic streaming. You don't need to know details about that, where the solutions inside the cells, they move around really quickly. It's very easy to see in plant cells, especially the ones growing in water. With animal cells, the good thing is because they don't have a cell wall, it's very easy to stain them with different dyes, especially the fluorescent ones that glow in the dark. I like to call them glow in the dark, but so when you look at them under the microscopes, under the microscope that allows you to look at the glow in the dark dyes, you can mix so many different colors. You can mix red and blue and get a purple color. You can mix red and green and get a yellow color. So you can get like a rainbow colored cell with animal cells because they're very easy to dye, very
Wow.
easy to look
That
at.
is like the art that you were talking about.
It's very artistic, yes. It's very, very artistic.
Well, we saw where you guys, was that one of your classes that did the designs with the growing and the culture? Like
No, that was Amy Doling's class. That was beautiful because what they did was they used bacteria that have depending on what they have inside, they are colored differently. Those are very cool too. Those are bacterial cells. But yeah, that's
Very
also different.
Campbell.
How many years did you have to go to college to study?
Oh my God, you don't even, I mean, no, it's fun. So I won't say. So for undergrad, I was an undergrad for like 3 years because I did my undergrad in India and that's a different system. I did my master's after that for 2 years, and I was in my PhD a total of 10 years. After
Wow. Long time after 12th grade, 10 more years. Okay, Liam.
What's your experiment that the results has interested you the most?
Like something surprising kind of thing.
Okay, so this is okay, I'm going to try let me try and simplify this. At one point, I used to work with fruit flies. Do you know what fruit flies are? They're these teeny tiny flies. You'll find them on bananas and apples if you just leave them out. They're everywhere. So we used to dissect fruit flies and we used to dissect the larvae, these teeny tiny maggots of these fruit flies. And so think about it. You have these small larvae that they look like worms, and then they develop into flies, right? Just like how a butterfly from a caterpillar, it forms a butterfly. So what's happening between, right? You have the pupae, or what else do you call it? I think the best thing is pupae, right? So from larva to an adult stage, you have these pupal stages. So we used to study what's happening in those pupal stages. And what we found is all the nerves, they have this covering around them. So imagine all the nerves. Think of all the nerves in your body. So from the larval stage to the adult stage, the entire covering just gets lost. So think, all these electrical wires that have those nice plastic coverings around them. So
Yeah.
from the baby stage to the adult stage, you have to remove those plastic wiring. And then you have to combine all the wires together and then form another plastic wiring. And we saw all of that. I could see all of that under the microscope.
That
So
is
I
so
dissected larva, I dissected pupa, I dissected adults. And in the pupa stage, I could see how that case around the wiring was removed and then it slowly got built up. You can see it happening under the microscope.
That sounds fascinating. So like structure to kind of more disorganized structure again.
Yeah, so you disorganize it and you organize it again. And that's what's happening even with adults, right? When y'all are young, and then when you go through your teenage phase, everything changes and your brain is getting restructured. And then by the time you become adults, you have a little bit more, oh, I know where I'm going. I mean, for the most part, I know what I want to do. So you should be kind to yourself when you're 11, 12, 13, because your brain is going through restructuring. Even flies go through that, frogs go through that, you will go through that too. So don't be, so be kind to yourself when you're going through that restructuring.
That's beautiful. Okay, how about we do two more questions and then I have a final question for her. Delaney?
What types of degrees do you have to do your job?
Okay, so like specific, like what was your undergrad degree? And
So the question is what were the degrees I had to do my job? Okay, so my undergrad degree was in just general biology. I always tell people for your undergrad degree, make it very uh, basic or fundamental. Make it very general. You don't need to specify because we don't know what we want to do, right? At the undergrad level. So I got a degree in biology for my undergrad. I got my master's degree in neuroscience. But for my PhD, I went back to cell and molecular biology because to understand neuroscience, you need to know a lot of cell biology, you need to know a lot of molecular biology. So I actually took a step back from neuroscience and I thought I want to study cells in general so that I can understand neuroscience better. So I was all over the place. But I enjoyed cell biology and molecular biology so much because I like working with DNA, I like working with cells. And I can at any point put that in neuroscience. I can any put time put that in genetics. So I got my PhD in cell and molecular biology. And after that, I worked as a researcher, both in like I told you, in the bioengineering lab, then in a neuroscience lab. Because I got a cell and molecular biology degree for my PhD, I could do all these other things.
Fascinating. Cooper, final question. Then we'll have two more.
How did you get discovered by NASA?
Oh, how'd you get to NASA?
Oh, when I was doing my PhD, I was helping out one of my colleagues. She had sent plants to space, and she needed help getting the data out really fast because she had to publish the work because NASA said, you have the data, it needs to be done quickly. So I started working with her on some plant cells to look at them under the microscope and to look at DNA and RNA. And we kept in touch since then. And because my work got published along with a NASA project, I wrote a grant last year. They said there was a there is an Iowa NASA organization, and I wrote a grant and I told them I have one semester off, which we call a sabbatical. I would like to come to NASA. I have got papers through NASA before, but now I want to use cells which are not plant cells that are different. They're called tetrahymena. Another story for another day. And they've never gone to space, and I think they're great as a model organism to go to space. So I wrote a grant, and because I already had some association with NASA, but they also thought this is a good project that's new and it hasn't been done in space before, that they said, okay, here's some money, and here's some money to travel to Kennedy's space to Cocoa Beach. So we'll fund your project. That's how and I'm working with the same friend I used to work with. She's doing a project on the side, and then she connected me with all these people. So never underestimate friendships in science as well.
I love that. And how important writing is. You had to do lots of writing to get these.
I had to do a lot of writing. I worked after hours to do the writing because I was also teaching and doing, all this other committee stuff. So it took a lot of time, but I enjoyed it. So
That's wonderful. Such important work. One final question, and some of them are disappointed they didn't get to ask theirs, but would it be
they
okay
can
if we
email
emailed
it to
you?
me. They can email it to me.
Okay, wonderful.
Or when I come back to Indianola, I can meet them somewhere.
Oh my gosh, we would love that.
Absolutely.
So final question, what advice would you give? These are fifth graders, so 10 and 11, that maybe want to be a scientist someday. What would you tell them to start doing now?
Okay. The one thing that I always like to tell everyone, because we tend to put a lot of pressure on ourselves to say, I want to become a scientist. And then we think of what is going to happen 10 years later. We think of things like, if I don't get this grade, then I can't become a scientist. Forget about all of that. Okay. If you want to be a scientist, focus on why you like science and just do that science. You don't have to worry about, do I like chemistry? Do I like physics? Do I like biology? If you like a biology project or if you like plants, work with plants. Forget about chemicals and forget about physics. If you like working with light or lenses and electricity, work with that. follow your passion. Don't worry about the outcome or what you need to do to become a doctor or an engineer or a scientist. Forget about all of that. Just do it because it's cool. Because you know what? Sometimes you might go in a different direction. I went in different directions all throughout my life, right? I did biology, then I went to neuroscience, and I went to cell biology, then I went to bioengineering. But here I am. And all of those things help me get to where I am right now. So just follow what your passion is right now. Don't focus on tomorrow. Don't focus on day after tomorrow.
That gives me shivers. That was amazing. Well, this was an absolute treat. What do we tell you, boys and girls? And we hope we get to see you in person sometime. That would be wonderful. And thank you for all you're
We'll be in
doing.
touch. We'll be in touch. I'll be back in August or September. I'll be definitely back in August in Indianola. So let me know if you want to meet up or get coffee together or something. I'll be there.
Wonderful. Thank you so
Bye,
much. Have a good day.
Have a good semester. Bye.
Bye.